I'd like to thank the organizer for this great opportunity to speak about the CANDID study. That's, as it has been mentioned a couple of times already, supported by industry partners, and I will go into more details about that. Can you see my slides?
We do.
Okay, perfect. Okay, good. So as it has already been shared with you with many details and nicely shared, I won't go into the description of the condition and the disease itself and the root of the disease, but I'd like to start with the very high unmet medical needs, which is very specific for the CDD population. Actually, right now we have standard anti-seizure drugs with limited efficacy aside the seizures. It was also very nicely explained by Scott Demarest in the video. And often, you have to give multiple drugs and with multiple dosages, which triggers or may trigger toxicity or some side effects. And that may impair cognitive aspects and domains, language, motor tone, motor control, and mood. Another strategy which is currently applied, that's the diets and ketogenic diets, for instance, neuromodulations with vagus nerve stimulations, or even more invasive with little evidence of efficacy, which is the corpus callosotomy. Having said that, so you will have a presentation following mine from Marinus, I believe, and ganaxolone was recently approved by the FDA as the very first CDD-specific anti-seizure drug. So that's a very good news, illustrating how dynamic the field is around the CDD research, preclinically, but also clinical research. And you have also heard about fenfluramine, which is now in phase 3, and Anna also described very well the landscape of the clinical research and where we are in terms of preclinical research for the different strategy involving the what we call disease-modifying therapies, because we try to, of course, improve the seizures, but with in mind improvement on other aspects and other domains which are beyond the seizures themselves. So we really need to have disease-modifying therapies such as gene therapies, for instance. But if we start embarking into the clinical trials with these gene therapies, expecting to have an effect and improvement on many other domains and aspects aside the seizures, we need to have outcome measures which are reliable, which are feasible in the population, and also recognized by the health authorities as good outcome measures to detect efficacy or to depict improvement. And that's the whole purpose of the CANDID study, but also the UO1 or ICCRN study, and I will go into more details at the end of my presentation. So I won't duplicate the very nice presentation from Anna, because that was really great. And at the last minute, I decided to include this in blue, which is more or less the terminology which was used by Anna. So the different strategies involving modulation of the genes. So you have the possibility to fix the genes or to add a gene into the brain. You also, and that's a work ongoing, you may want to replace or to add the protein itself, which is produced by the gene, CDKL5 genes. And you may want to include or to inject this protein into the brain directly. And you have also the possibility to reactivate or to read the inactive chromosome, the X chromosome, where you have the copy of the CDKL5. So these are the different strategy, and with the presentation from Intragenics, you have understood that we are really close to reach the clinical trials and the clinical phase. So we really need to have something done quickly to make sure that the outcome measures will be accurate and reliable for starting these studies. To prepare this, we need actually what I call here the natural history study. So here, of course, I will detail the CANDID study, but there is another parallel effort, which is US-based with the your one study helped with the help of the IFCR team. In our case, since we are targeting US but also ex-US countries with many different languages, we need also to have a strong connection with the patient advocacy groups which are in each country. So that's why I wanted to highlight the CDKL5 Alliance here for helping us starting the operational aspects for the CANDID study itself. But to prepare the whole development, what the industry needs, and Heather started to explain those different needs, you need to also prepare the biomarkers landscape. Here, I have split between the fluid biomarker, which are like blood-based biomarkers, but you have also functional biomarkers, such as the EEG or sleep architectures you may want to evaluate. We have good hints from the preclinical experiments that these might be very nice and good biomarkers. These biomarkers will help actually to be super efficient in clinical trials and to identify whether these biomarkers could help us adjusting the doses, for instance, or detecting improvement even before functional improvement. And the last piece, which is really critical, that the patient registry itself, which has been described and detailed by Dan and Femida earlier today. And that's very important in the industry. They call this real world data. It's simply a way to gather very important data on how the families deal with the daily aspects and the impact of the disease on a daily basis, on other elements that you can collect through or from a natural history study. So I wanted to explain how we ended up with the CANDID study. The Loulou Foundation wanted, I'm sorry, I don't know why I have that. Okay, there you go. So yeah, I really wanted to explain how we ended up with the CANDID study itself. It's not like a study, coming out from a hat like that. It really needs a lot of work up front. And it all started with what we call patient advocacy groups, meeting with the FDA, more specifically here, the one we call PFDD meeting for patient-focused drug development. That's an opportunity for the whole community to expose the FDA with a new disease, in our case, the rare disease and the CDD itself. And that's the first time you interact with the health authorities to explain the family's views, how it goes, what is really important for them to be treated, beside the seizures again. And that was basically held in November 2019. But then the second stage, which was extremely important, in October 2020, was issued an ICD-10 definition or code. It's basically an official definition of the study, the disease itself. It allows you to have the disease itself recognized by the health authorities, but also the industry and all the community to have a clear understanding of what we would like to really characterize and to measure later on in drug development, for instance. In February 2021, we went back to the FDA to present and discuss our CANDID study. That's a very specific, let's say, informal meeting, but it's a really nice way to discuss and start discussing scientific aspects with the FDA officers on the natural history study as an example, and in our case that was the CANDID study. It's also a way to discuss the design of the study, what we're going to measure, if they agree with our selections of outcome measures and things like that. And of course, that's also maybe a first way to interact with the officers on potential phase 1 clinical trials when we're going to start testing a gene therapy or a drug in humans, and to see how we could deal with the different type of disease, especially in these rare conditions. And potentially with gene therapy, you have invasive administration of the drug, which is very different from tablets or caps. So that's also a way to start discussing with the health authorities. The consortium itself was public and set up in October 2021, and we officially started the study December last year. Supported by 7 companies, Ultragenics, of course, Amicus, Marinus, Biogen, LH Bio, UCB Sogenics, and PTC. And they were all, part of this CPI meeting and discussion with the FDA, but also they were all involved in the discussion of the design of the study, selection of the scales. And that was a team effort, I would say, and Loulou Foundation is really there to coordinate the study and to have it implemented and done with all the operational aspects. What we also wanted to implement, that's the quality standards which are used in the industry, so that we are mimicking more or less what we're going to have in clinical trials with a gene therapy or with a drug later on. So with this in mind, we had to select a CRO, which is a company helping us operationalizing the study, meaning that we select the sites, we select the country, we are making sure that the quality standard are really in place and consistent with what is expected by the industry and the health authorities. Cogstate is another company, but specialized into outcome measures. So that's very important in our case with this study, where we're going to test different type of scales and questionnaires. The Epilepsy Consortium, Study Consortium, is another entity helping us to standardize the way we collect the seizure and more specifically the seizure types. This is very important, not too much in this type of study, but later on to prepare the clinical trials and to make sure that we have all the sites, all the countries, all the PIs collecting the seizure and assigning the seizure types the same way. And Pulse InfoFrame was also very important to implement what we call the global unique identifier. It's basically assigning a subject number, a participant number that could be used in different contexts, different studies, so that if we have only one number for one particular patient, we can link all these data together and make the best out of the different studies which are ongoing, and that includes also the patient registry. So based on the clinical presentations, which has been detailed in a paper issued in 2019 by Olson et al., we have a clear understanding of the clinical aspects, symptoms, which are really key and specific to the CDD populations. And also it helps us together with what we call a disease concept model to identify the domains where we need to give the priority for these different scales and for this type of study, natural history study. So with this in hand, we could identify the different domains we wanted to explore, and these domains that needs to be implemented in future clinical trials. And that's why here on the right-hand side of the slide, you see we have quality of life, sleep quality, cognitions, corticovisual impairment, behavior, motor function, seizure types, of course, and frequency. All together, they need to be evaluated throughout the duration of the study, which is planned for now to be 3-year studies with, you see what I called home visits. It's basically nothing else than visits done from home, over the phone, for instance, with the investigators or the clinicians, or with the raters, the neuropsychologists administering the scales, and other clinical visits. So what we call the baseline visits, the first one, and then 6 months later, a year later, a year and a half, 2 years, and 3 years. All these measures and domains explore have different ways of being measured and evaluated. Most of these domains are basically explored via questionnaires, and this is what you see in green, or interviews. And that's why we can basically design the study with several home visits and visits done over the phone. We have direct measurements, meaning that this is a psychologist, raters administering a specific scales to the kids, to the participants, to evaluate the cognitions, to evaluate the motor function, for instance. And we have also the feedback and input from the investigators and the clinical experts themselves, giving their input on, how the participants' functioning level is basically throughout the 3 years. So we can also evaluate, the trajectories of some of these capacities. But I need to explain, because here I had a slide detailing all the scales, but the most important thing in the CANDID study is that we are testing already known scales, already known questionnaires, which has been used in other clinical trial settings with similar conditions, also with people very impaired, either on the motor aspects and motor functions or in terms of cognition. So we know already that these scales and questionnaires were accepted by the health authorities. We just want to test their suitability and their feasibility in our population with CDD throughout the different age ranges that we had selected for the study. So that's the primary objective of the study, to evaluate the feasibility, suitability, and the trajectory over time of these different scales. We would like also to evaluate the influence of the age, because we're going to go from birth until 55 years, and also the gender effect that may, impact some of the outcome measures and the results. We want to evaluate this impact. We are expecting, for instance, to have maybe the females and the girls being less impaired than the males, because of the the fact that you have two chromosome X, and sometimes the other chromosome helps diminishing the impairment in the populations. That's something we would like to evaluate with these different scales as well. And I have mentioned that also the trajectory is important to be evaluated together with the impact maybe of the seizure severity, and if it has any correlations on the different results we're going to collect. The study populations we would like to enroll into this study, 100, we are targeting for approximately 100 participants from US, Canada, UK. So these English-speaking countries will be the first countries enrolling participants. And it's I'm pleased to say that it's a question of weeks now, because all our US, Canadian, and UK sites have been identified, and we are basically discussing or submitting our protocol to the ethics committee and IRBs at the moment. And then it's going to be followed by France, Spain, Italy, Germany, and potentially UAE sites. Why it's a stepwise approach like that? It's simply because some of the scales and questionnaire needs to be translated, and it takes time. It takes sometimes almost a year. So we are expecting to have France, Spain, Italy, Germany, maybe starting enrollment in November this year. And the inclusion criteria or non-inclusion criteria are very large, basically because we are looking for documented diagnosis of CDD with pathogenic or likely pathogenic CDKL5 variants, and participants can be aged as old as 55. I know it seems to be old, but we have a good example, including in France. I have met families as a girl with age 45, for instance. Yeah, we are confident to have some adults in this population and in this study, and that's a very interesting population to evaluate, since the diagnosis criteria were issued 2 years ago. I know that we may have difficulties to identify the adult patients for this study. Okay, I'd like to close the presentation with a couple of words on the other non-drug study or observational study. which is the Year 1 study, NIH granted study. And why doing this is simply because for the first site starting the study and starting to enroll participants, it will be American sites. And I know that from the Center of Excellence, they are already involved in the ICCRN study. But actually, that's a very nice opportunity here. There is no competitions really. The only competition is basically for enrollment and the family burden aspects. But this is a luxury. And I can tell you that in other rare diseases I've been involved in, I've never seen that before. Previously, I was working in the industry. And why it's such a luxury to have these two natural history studies in parallel, I try to explain that. The first thing is that we have two different strategies. The first one I see for the candidate study itself is to evaluate the suitability of already known scales and validated tools validated in similar conditions. The other strategy, which is followed by the ICCRN study, is to generate and validate clinical outcome measures results from a brand new scale specifically designed for the CDD clinical trials. So basically what it means that we have two different strategies in parallel, but each study can help the other one. Because the candidate study, for instance, will benefit from the expertise from the Center of Excellence sites in the US, of course, but not only. We will get probably EEG data, evoked potentials data, CGI, that's another scales or hand function data that we don't have in our candidate study, but which is implemented in the ICCRN study. So if we have one participant involved in both studies, we can connect this data together, and it's going to be really, really nice to have this type of information and informing our results and the other way around, because the ICCRN study itself will have a way to further validate the CDD severity assessment. This is the brand new scale which is being developed. And of course, to correlate or look into these results against the results that we're going to have from the candidate study, right? So that's really a nice thing to have all these two studies together, and we work closely together. I'm also involved in the advisory team for the ICCRN. I'm following on a monthly basis, and I give updates about the candidate study, and we try to have these two efforts being done together. The protocol for the candidate study has been also written in a way that we are flexible enough not to increase the burden to the families. So if we have like two scales administered at the same time, of course it won't be we won't administer twice the same scales or questionnaire in both studies.